do that by talking about this resource that I run called the BioImage Archive. But just before I do that, I'm going to take one step back to think a little bit about why you should be thinking about archiving your data at all. So Harald and I are telling you almost the how and the where you can archive your image data, where you can publish it publicly. But why should you do that? There are a lot of reasons, but I'm going to pick on 4 particularly. And the first and perhaps the biggest is that biology is a data-driven discipline, and to make science work, to make biological science work, we need access to those data, particularly when it comes to both reproducibility of results and really sort of methods development and pushing forward new science. And I'll talk a little bit about this more on the next slide. But there are other reasons to want to archive data that are perhaps a bit more personal. So one is that it gives you publicity for your work, particularly if you're doing something that is, say, methods driven in biological imaging. If you have some really nice new technology, or you've developed a new sort of fixation method that gives you particularly beautiful images, it gives you a way to share that with the community that's independent of the parts of the story that might go into a publication. Another reason is that actually managing your image data, particularly in the long term, is quite challenging. You may well find that you know where your data are now, but in 5 years' time, when you're looking back to think, how did I do that experiment? What was it we captured? Those questions can be difficult to answer, particularly since as scientists, we tend to move between institutions quite often. And if your data are trapped in some institutional repository that you no longer have access to, that can be a problem. And final reason I'll touch on now, which is really linked to the first reason, is that increasingly you will be sort of initially gently and over time more firmly pushed to archive your image data, as in the sort of genomics and structural biology worlds has become very much standard. And that pressure is coming from publishers, from journals, it's coming from funders who want data management plans explaining how you'll make the data that they are funding openly available, and it will also come from individual institutions. So there were good reasons to do this, but how? So I'll tell you about the BioImage Archive, and this, as Harald introduced, is the newest image data archival resource at EMBL-EBI, with a very, very broad scope. So we aim to make bioimaging data from any modality, so light microscopy, say 2D electron microscopy, but atomic force microscopy, really almost anything you can think of, available to the world, and really with 4 aims. And one of those is reproducibility. So I mentioned that science, biological science is sort of built on data, and image data has been a huge part of biological progress for literally for centuries. But it's increasingly true that the path from the data to the sort of understanding and confirming or disproving hypothesis depends on close analysis of that data, often through sort of numerical and statistical techniques. And so to really check whether those hypotheses, whether the data is showing what somebody is claiming that they're showing, it's impossible to verify those claims without having access to the data that we use to make them. So we need to provide a way to do that. We also need to provide a way to host and share what we sometimes call reference data sets. So data sets that are part of large community efforts to really understand and image in detail some biological system or area. So cell atlases are a common sort of source of reference data, and those are hugely time and therefore effectively money intensive projects. So we owe it to the scientific community to make those as available as possible for as long as possible. And the remaining 2 reasons are actually about really new science, and new science from existing data tends to come to some extent in 2 ways. Firstly, in methods development, so image analysis techniques, and particularly modern sort of machine learning and AI techniques are very, very data hungry. They need large corpuses of reference data to train models, but also to test on. So we need ways to make data available for that reason. But also particularly sort of looking back at areas in science or biology where there have been histories of good data sharing, like structural biology and sort of sequence genomics. Often new scientific questions can be answered from existing data. And so again, without open access to that data, we can't do that. So we're trying to help with this process, and we're trying to do that by taking bioimaging data really from imaging at any scale for molecules to organisms, and we have examples of those already in the BioImage Archive, and we'll take any format of data as well. Really our mission is to make sure that we can host the data. Our main constraints are firstly that data has to be either related to results in a publication or show clear value beyond a single experiment. So that is to some extent the reference data sets I was talking about. And the reason for that is we're not trying to be the sort of image data management platform for the world, because that's just not scalable. We can't manage that. It has to be something that has, in a sense, already proven that it's useful. And we also can't take patient identifiable medical data like clinical data, because that requires quite a complex sort of governance and anonymization infrastructure, which we're not currently, at least, in a position to support. And I should sort of make one concept more clear before I start to talk in detail, in a little bit more detail, about how the BioImage Archive works. And that, again, is something that comes from the genomics world, and that's a separation of data resources into primary archives. And primary archives are really there to be very broad-based and to take data, faithfully store that data in as close to its raw format as possible, and keep a certain minimal level of metadata for that data. I'll talk more about metadata later. And then on that can build what are sometimes called added value databases. So these are data resources that have expert curation. So they'll often, as EMPIAR does, have staff with sort of deep scientific experience in a particular domain, and that can use that experience to really build on those data. And to some extent, EMPIAR and IDR, which I'll talk about in a moment, are added value databases, although actually this is really a broad spectrum, and EMPIAR is also sort of primarily archiving that data, but it's building a little bit more on top of it than some of the things we do in the BioImage Archive. And if you're interested in this model, then there is a sort of good description of how it applies to image data in the paper that I put on this slide. And in response to one of the questions in chat, we'll make all of the slides available in PDF form after the talks. So I mentioned the IDR. What's the IDR? It's the image data resource. And this is a data platform that was developed by the Open Microscopy Environment team, who are sort of very big part of the world of biological image data management, together with EMBL-EBI, and it launched a few years ago, I think in 2016. And it holds curated reference data sets. So it holds cell and tissue imaging from data sets that are extremely complete, so they're extensive, and they look at particular types of sort of biological dynamics where you're doing studies over a large number of genes or small molecules or something like that. But it's hosted actually alongside both the BioImage Archive and EMPIAR in EMBL-EBI resources, which is good because it allows us to share data in the back end. Final resource I'm going to mention briefly, because this sometimes comes up in questions and you might have heard about it, is another EBI resource called BioStudies. And BioStudies is a data resource designed to do two things. Firstly, to allow linking together of lots of different types of data. And it's one of the reasons why there is a picture of a link in this screenshot here. And also to sort of take data that can't fit into a more specialized resource at the EBI, which is the position that general imaging data was in for a long time. And because of that, we built the BioImage Archive on top of BioStudies. So you may possibly previously have been using BioStudies to host image data. And now we point you towards the BioImage Archive instead, but under the scenes, BioStudies is actually providing the platform on which the BioImage Archive builds. So suppose I've convinced you and you would like to submit data to the BioImage Archive. How does that process work? I'll go over it at a very high level rather than details. And if you want more on the details, as you'll need if you actually come to do a submission, there are extensive instructions on our webpage, and you're given more help and guidance through the tools that you'll use. Plus, you can get in touch with us. There's links to sort of help references and email addresses. But at a high level, the process works like this. You organize your data, so you make sure that you know what you're archiving, because often when you do an imaging experiment and then some analysis, you'll build up a large collection of data. And to begin with, you tidy that up and organize it and work out what is a sort of coherent set of data to describe your experiment that should go into the archive. Then you'll need to register an account with us and then transfer data over to EBI. And that's the part that can take potentially the most time. If you're archiving smaller data sets, so up to a few tens of terabytes, you can expect to do this in a day. If it's into the hundreds of gigabytes into terabytes range, you will need to allow potentially several days. I know that for EMPIAR, for the very large entries, which are potentially hundreds of terabytes, at that point you need to allow weeks to transfer data. So it is something to think about in advance. It can be hard to accommodate I must publish tomorrow deadline if you have a lot of data. And then finally, you fill in some metadata about your data. So you need to describe what it is you're archiving so that other people can understand it. And if you want to find out more about this process, there is a link down here to an article which is actually focused on correlative deposition, but really includes sort of standard deposition within that. That last stage, the filling in the metadata, you do through an interactive web form, which we provide. So you go through different types of description and fill in different fields. And if you need to go away and look something up and come back, you can do that. The progress is saved throughout the process. And then when you finish and you submit your data, it will be sort of a permanent copy is made on our servers and storage systems, but you can decide the point at which it will be made visible to the public. So for example, if you want to check everything looks okay, that's fine. If you want to share a private link with journals or referees, you can also do that. And you can also, for example, come back and edit the publication details if you have a preprint, but you want to associate a full manuscript later on. I've mentioned correlative imaging, and correlative imaging might come to mind if you're thinking, okay, well, there's a resource for EMPIAR for 3D EM data and data underlying 3D structures, but I've got data that's also light microscopy data. What do I do? Well, we provide a protocol, which is actually described in more detail in the paper that I linked a couple of slides back, about how to deposit that. Really, the way it works is that your light microscopy data goes into the BioImage archive. Your 3D EM or perhaps X-ray, in this example, data go into EMPIAR, and then we can link the two together so that we can make sure that these data sets are associated. But the BioImage archive also takes the transform side of that. So if, for example, you have descriptions of fiducials, or you have transforms that map both of these data sets into a shared set of 3D coordinates, which is the part you need in this illustrative example to get the two data sets aligned, then those go into the BioImage archive. And again, this is described in more detail in the publication. And then you end up with something like this. So there'll be data sets in the BioImage archive, and there'll be data sets in EMPIAR, and there'll be these links which join the two together. And as Harald mentioned, as we develop both of the archives, there'll be more ability to cross-visualize these data sets in interesting ways, and allow further downstream analysis of them. So a very, very brief tour of types of data we have. We have what you might think of as the sort of traditional, almost light microscopy data set. So this is a protein localization study on human cells, and you can download both individual images. You can download the whole data set if you're interested. You can just look at the metadata. You can sort of search and filter for certain files, and I'll talk a little bit more about that in a moment. But we have plant data as well. We have screening data. So this is a high content screening experiment looking at cytotoxicity on SARS-CoV-2, so this was quite topical. And one of the things that we allow, and we allow it for any data set, but it's particularly relevant for screening and high throughput data sets, is that you can provide specific metadata for each image in quite a flexible way. So for example, for these type of compound screen, what's really important for those who are looking at the data to know is what compound treated each well. So these experiments have large multi-well plates, and you treat parts of your cell line with different compounds. And here we have both the names of the compounds, and we have formulae, and we have sort of references to other databases as well. And you can search and filter by these things. So if you're particularly interested in effects on one compound, you can dig deeper into this data. So the system for providing this metadata is very flexible, because if, for example, you're doing a genetic knockout screen instead, it would be a different type of metadata. But beyond these sort of more almost standard data sets, I mentioned that we're trying to cover a very wide range of image modalities, and we've actually found that we are capturing a wide range of those modalities. So we have ultrasound data, we have micro CT data, we have photoacoustic imaging data, and these are less common than light microscopy or 2D EM, but they are there. So if you're interested in sort of more unusual imaging techniques, then we might be a good starting point if you want to come and look for some sample data for those types of imaging. Last note on growth, we're a very comparatively young resource. So the BioImage Archive started in 2019. I mentioned before that it was previously possible to deposit imaging data into biostudies, which is why this graph goes a little bit back in time before the birth of the BioImage Archive, just to show the growth. And these are the cumulative number of data sets that we have. And since the archive launched, growth has been pretty rapid. And I think especially at the beginning of this year, we've noticed that it's growing even faster. So although the absolute number of data sets we have isn't enormous, it is growing very fast. Last section of the talk, where are we going in the future? Well, I've mentioned metadata several times. And as part of the process of setting up the BioImage Archive, I think we realized at the EBI that we would need a way to describe biological imaging experiments that could really cross over all of these different modalities and types of imaging that we're interested in. And that's a metadata schema, or perhaps a set of metadata schema, metadata sort of data about data. And to do that, the EBI got different working groups from different imaging modalities together to try and agree on a common standard for describing imaging experiments. And the result was something called EMBL-EBI, which stands for Recommended Metadata for Biological Images. And this is really a framework for describing the different metadata components that we think are needed in order to understand what was done in a biological imaging experiment. And really these go from the broad, so for example, study is expected to include why you were doing your imaging experiment, who was doing it, so the study authors, what the sort of the timelines for your experiment, perhaps when you released your data were, and then as you go round the model, it gets more specific. So the sample is what were you imaging? How did you prepare it? How did the actual acquisition process work? What perhaps were your microscope settings? What was the data you produced? What are the formats and sizes and types of those data? And then where it's relevant, for example, for image correlation, which we've talked about quite a little bit, that correlative data I mentioned that you need in order to be able to transform data, the different modalities of data to line up. But also if you have analyzed data, so if you have, for example, you've done cell counts or cell sizes, we can accommodate those in the metadata model as well. And we're currently implementing EMBL-EBI as the metadata model for the BioImage Archive, and we're actually hoping over time that this will become standard for image data archives, so that there's more ability to interoperate and understand how our data work. And then again, Gerard mentioned that the requirement, I think, particularly for EMPIAR, coming from the growth into 3D volume EM imaging, to have file formats that are really designed for hosting three-dimensional data in what we sometimes describe as a kind of cloud way or streaming way or random access way, ways that basically allow selection of components of the data without having to just download an entire data set and work with it locally, which for multi-terabyte data sets is not practical for people. You want to be able to look at individual planes in an image or subvolumes or parts of a time series. And there's some really nice work going on in the community on formats that are designed to do this. And one of the leading candidates is something called OME-NGFF, which stands for Next Generation File Format. Another big advantage of doing that for the BioImage Archive is that there's also a rich ecosystem of viewing and visualization tools, like the one I have illustrated here called Visar, that will enable us to provide better visualization of the data that we've got, either in browser so that you can browse through the details of our data sets, or if you want to do that locally, but with the data hosted on our servers. And finally, we are aware, because in a sense we've seen the success and growth of EMPIAR, that in the future managing the potentially enormous volumes of imaging data is going to be a challenge. So we're thinking a little bit about how that will work in terms of how and where we store different components of an imaging experiment. And then the last thing is that we talked a bit about correlative imaging, and correlative imaging is sort of multimodal. It's a multimodal experiment where the modes are both imaging data. But you can go beyond that in a sense. You can do experiments where you have both imaging and, say, genomic data. And one nice example of that is spatial genomics. So this very, very beautiful image in the picture, in the middle, sorry, which comes, I think, from the Sanger Institute. Each dot on this is a different RNA transcript. So there's 12 different probes here corresponding to different transcripts. And to really understand and interpret this, you need both the spatial information, which is what the image is giving you, but you also need the genomic information. The genomic information is often in genomic specialized data resources, like some of the ones that I've listed on the right-hand side. But we want to be able to integrate those to really sort of give the best level of understanding possible for that type of data. So we're only really at the beginnings there, but that's something that we're working on. And then to make sure we've got time for some questions, I'll finish here just by thanking several people. So the group on the left are my immediate team, Colin Livy down the bottom, and some of my many very supportive colleagues at the EBI, because this has very much been a group effort. And then the group on the top right are our ecosystem sort of pre-advisory group. And I should absolutely thank our funders, both sort of current and past funders, because they've been extremely supportive throughout the project. And finally, thank you to all of you very much for listening. So